Alrighty, good morning there, Biology 30s. Today we're going to start talking about meiosis and sexual reproduction. So we've already learned about mitosis. So we've got some similarities and differences that we're going to clue into here. Now, again, these notes are from AP Biology teacher named Kim Foglia. And in your textbook, this stuff is actually chapter 16 on meiosis and sexual reproduction. So, when we talk about cell division, mitosis is asexual. We're producing identical daughter cells. We're making exact copies, clones, identical copies. And so each cell that is formed has the same DNA, same chromosomes, same genetic information. The purpose of doing this is for growth, repair, and to replace dead or dying cells. So again, identical copies, but there's no genetic diversity when this occurs. So examples of asexual reproduction include a lot of single-celled organisms. This is their only method of undergoing reproduction. So one cell becomes two. And again, there's no genetic diversity. This is a disadvantage of asexual reproduction. This diagram or picture here, I guess, shows that here is an example of asexual reproduction called budding. So this little mini-me, this hydra, this is animal that is going to bud off from the parent. So it just grows off the side. It's an identical copy. It's quick. There's less energy required. You don't need two parents of the opposite sex. So those are some advantages. But again, there is no genetic diversity when this organism is just making a copy of itself. So another example is budding in yeast. And again, this is just simple mitosis. You can see a yeast cell, and here's its new little mini-me just growing off of the side of it. Each of these is going to produce an identical copy, right? It's going to be just like the parent. It's the same. One cell is becoming two. It's just mitosis. So yeast, amoeba, any sort of single-celled organism are just going to reproduce very simply just by mitosis. But what about the rest of us? What about humans? What about animals? What about plants? What if we want to reproduce? So if we just take a 46 cell like human being and 46 cell egg and sperm, and when you put them together, you'd have too many chromosomes in the zygote. So we can't make sperm and egg by mitosis because there'd be too many chromosomes for that organism to survive, right? It wouldn't be human. We need to have those 46 chromosomes. So how do we do this? How are we going to have the right number of chromosomes? Well, in any female organism, human organism, we have 23 chromosomes from dad and 23 chromosomes from mom come together at fertilization to make a zygote. And then as that zygote divides, every cell in your body now has those 46 chromosomes. So if you were to take a look at your chromosomes, and this is called a karyotype, and this is of a female. And how do I know it's a female? Well, I look down here at this 23rd pair of chromosomes, and I see there's two X's here, X and an X, and there's no Y. So this has to be a female. The chromosomes 1 through 22, these are the autosomes, the body chromosomes, and these determine things like your hair color and eye color and whether or not you're going to have Huntington disease. And so those are your body chromosomes. And that 23rd pair, that's the sex chromosomes. And so if you're XX, you are a female. You want to compare that to the chromosomes in a male. And what's the difference? Well, the difference is down here at the bottom. You have the XY. And so you'll notice that X chromosome, rather large chromosome here, and the Y is a little tiny chromosome. So that's our 23rd pair of chromosomes, the sex chromosomes. Okay, so XY for male, this is a normal male karyotype, and a normal female is going to have an XX as the 23rd pair. Moving right along, to make sperm and eggs, obviously then we have to reduce the number of chromosomes. We have to take a diploid cell, like a body cell, 2N, and reduce the number to haploid. That was how females make eggs. They undergo in their ovaries meiosis. This is oogenesis, the production of eggs. Males undergo spermatogenesis, and they're diploid 2n46 chromosome cells. They have to reduce the chromosome number, half it, down to 23, so that when the sperm and egg come together at fertilization, we join those gametes together. Now the zygote is going to have the correct number of chromosomes, and that diploid 2n number is going to be 46. So meiosis, when you look at any alternation of processes, the sperm and eggs are always haploid in humans, and zygotes are always diploid. And of course, meiosis reduces the chromosome number, right, in order to make the haploid gametes, the sperm and egg cells. And then the fertilization is going to bring that number back up to the diploid. It restores the ploidy, it restores the chromosome number. So here's a new word for us when we are looking at meiosis. We are talking about homologous chromosomes, or tetrads, or homologous pairs of chromosomes. So these have similar information to one another, but half, remember, comes from dad. That's our paternal homologue, and half comes from mom. That is our maternal homologue. All right. So they are paired up as tetrads in meiosis 1. So again, sexual reproduction, we are taking an egg from mom and a sperm from dad. They're both haploid. They come together at fertilization, and you get a diploid organism. This is the zygote. This was you at one point in time. And you now have the full chromosome complement. You have a diploid number of chromosomes in humans, that is 46. So what might be interesting is you might be asked to, maybe you have like a horse or something, and let's say that the number of chromosomes in a horse, maybe their 2n number is, say, 96. And then you might be asked, well, then what would be the number of chromosomes in a sperm? So then you would have to say, it's going to be half of that number of 96. And then you would have to calculate out on your calculator, well, what is half of 96? And that would tell you the number of chromosomes in a haploid sperm or egg. So 48 would be the number in that horse or whatever the organism is. Okay, and vice versa. If you're given the n number, then you just double it to make the 2n number if you're asked for the ploidy of an organism. So again, this whole process of meiosis is doing the exact opposite of what we just saw at fertilization. It is reducing the chromosome number by half. It is setting up gametes, and that is the purpose, right? To make gametes, to reduce the chromosome number, and to get these cells that then are going to be able to make the next generation of offspring. So we're going from 2n diploid to haploid. Now, some people maybe remember that as haploid, right? It's half the amount of chromosomes. So you go from a normal 2n46 chromosomes down to 23, and that's haploid. It has half the amount of chromosomes. Okay, so there are two sets of divisions in meiosis. The first round of division is called meiosis 1, and the second round of division, meiosis 2. DNA replication has to occur before meiosis 1. The first division of meiosis is going to separate the homologous pairs into chromosomes, and then the second division is going to separate the chromosomes into the chromatids. So before meiosis even occurs, we have to make sure that each of the daughter cells gets enough DNA. So we have to go through an interphase. We know that S phase of interphase is the stage in which we are going to make an extra copy of the DNA. We're replicating or duplicating or copying the DNA. That way, then when we undergo meiosis, then all of the cells that are produced will have enough genetic information, have enough DNA. Okay, so I'm just going to stop there. Our next video, meiosis 2, is going to talk more in detail about the stages of meiosis and do a bit of comparison between mitosis and meiosis. And we will also have to talk about the important events in diversity. Okay, so be waiting for video number 2 on meiosis. In the meantime, if you have any questions, michelle.ledeen at ghsd75.ca. Talk to you later.